Good evening, ladies and gentlemen. Welcome to the University of Melbourne and to the second ICT for Life Sciences Forum presentation for 2012. My name is Luan Ismail. I'm the convener of the forum, and I'm very pleased so many of you are here this evening. The ICT for Life Sciences Forum provides a unique opportunity for the community in Melbourne to hear and meet local and international experts share with us the exciting world of the new biology, where biology is converging with physics, mathematics, computing and engineering, and the least of which is its role in giving personalised medicine real meaning. And following on from our first presentation in March, we are again serving you a diet of systems biology this evening. It's no exaggeration to describe this evening's presenter, Professor Michael Stumpf, as one of the world's leading scientists who is building a vision of the role of systems biology in health and biology in general. Michael was trained in theoretical physics at the University of Tübingen and completed his PhD in statistical physics at Oxford University, where he was a fellow in mathematical biology in the Department of Zoology, and currently holds the chair of theoretical systems biology at Imperial College London. Given his background, he is well positioned to make sense of the powerful forces in which ICT and biology are intersecting. Please give a warm welcome to Professor Michael Stumpf.
Thank you very much, Luan, and thank you very much for coming here. It's probably the only lecture theatre I've ever been to which actually looks really nice if nobody's sitting in any of the seats, because it's so nice and colourful, but it's even better if some people are here, so thank you very much for coming. So I will talk a little bit about activities that are now ongoing at Imperial College, where we are trying to bring cutting-edge physical sciences, engineering sciences into a clinical setting. And before I will discuss this, it's useful to have a slightly sort of the luxury of a historical perspective. And it is important to remember that until the middle of the last century, medicine has probably killed more people than it cured. And sometimes when doctors decide it's a good idea to wash their hands as they go from one ward to another ward or between surgeries, it starts to revert. Here is an image of the Royal London Hospital for Homeopathy, which during the 19th century was pretty much the safest place in London. So fewer people were killed in that hospital than anywhere else in London, including all the other hospitals, including also the hospitals that are associated now with Imperial College. If we look less flippantly at the advances that medicine has made over the past, especially 100 years, then we will see that in this list, if you can make it out, the main results that have really benefited a large proportion of humanity in terms of reducing morbidity and mortality were not necessarily cures, but preventative measures. There were various vaccines that have become very famous, and one of the most important studies, for example, was the British doctor study published in 1956, which is largely responsible for smoking being banned across large parts of the industrialized world today, which probably had a much larger effect on reducing morbidity in Western countries than most of the other things that we have here, plus generally getting rid of infectious disease. And we would like to do things better now. What we would like to do is try to identify a set of therapeutic interactions that are targeted at an individual patient rather than sort of giving everyone an aspirin. And we would like to understand the mechanisms of disease and of drug action. And here we have to make a compromise. We either understand the mechanisms by which these diseases work, or we try to identify the best therapeutic intervention. And those two things are not necessarily closely related. So you can do one thing without the other, and I will try to touch upon both aspects of the rest of this talk. So briefly, in personalized medicine, we are trying to customize the healthcare and the treatment at an individual patient through the use of genetics, molecular diagnostics, and general information about the environment, lifestyle, and family history. So the last three things, environment, lifestyle, and family history, have already been part of any diagnosis for the past century or so. Only the last 50 or so years, if you're so slightly optimistic, have given us access to genetic data. And really only since Landstein found the ABO blood system, and then more recently in the last sort of 10, 15 years, were we able to use advanced molecular diagnostics. And I will come back to some of those in the future developments later on. So, but that is what we understand by personalized medicine. And I will discuss the challenges, which is a nice cop-out because I don't have to provide solutions. I will try to provide a couple of solutions which you might find boring because they're largely in the statistical realm, which is really important in this field. Anyway, so what do we want to do? Ideally, and that is the sort of thing when I started to become dissatisfied with physics and wanted to move to biology, I sort of fell for this paradigm or this notion that from genome sequence, we might be able to identify the causal mechanism of disease. I persuaded the Wellcome Trust. I was one of many people who persuaded the Wellcome Trust that this was worthwhile investing in, and got a fellowship and so on, and left physics. And in a way, it is a bit oversimplistic, as it turned out, because there are other things such as environment and the lifestyle. I think it's useful to distinguish between those two things and have a simple functional distinction where you say lifestyle is everything that you can do and stop under your own volition. Smoking, taking up exercise, eating less or more are aspects of that. Environment might be harder to change. If you come from, like I do, from the Bavarian forest, then you're automatically exposed to higher environmental radiation. But also we have to keep in mind that for any cell inside your body, you will have 10 microbial cells lying inside your gut. So in our own bodies, we're outnumbered 10 to 1 by microbes. These I would put under the environment as well, because it's very, very hard to change your microbial makeup. Your gut flora is hard to change. So that's why I think it's useful to make this distinction between lifestyle and environment. And we know now that for many diseases, especially metabolic disorders, but probably also some autoimmune disorders, the environment, the gut flora, our microbial passengers in our bodies have a role to play. But these things do not directly affect the disease, and we don't identify disease, we identify symptoms. Doctors identify symptoms and then try to see what was the disease and what was the causal mechanism that gave rise to the disease. So symptoms can range from every disease that kills you will start like a common cold. So if your nose starts if you start to sniffle your nose, that is one symptom. It's not very specific, but you can have more specific symptoms and so on. And you would like to identify the disease based on the symptoms. But between here, the genome and the disease is a molecular phenotype. So it's the proteins, the protein-protein interactions, the protein-DNA interactions, RNA interactions with proteins and DNA and metabolic interactions, which modulate what you inherit through the genome, together with environmental and lifestyle cues into something which can or cannot give rise to a disease. And we will focus on this aspect here for reasons that will come later. And finally, there's a small interaction here between your genome sequence and your lifestyle. Certain genomic characteristics might make you more susceptible to certain silly ways of leading your life, and certain types of your genome sequence might also predispose you to certain types of gut flora, for example. So there are some interactions here which I won't focus on, but I think it's important to keep in mind that these things will complicate matters quite enormously. So one of the starting points for our studies at Imperial College was, what is better to identify which disease or which causal mechanisms we have? Is it the genetic sequence or is it the symptoms? And here, and I won't go very far into that thing, we have started to employ with reasonable success an information theoretic framework, which just is a more generalized way of looking at correlation. So you have a disease D, and you have either a symptom or a genetic allele X that might predispose you to a certain disease, and you try to calculate how much information do you gather about the disease phenotype if you know, for example, the state of a gene or a symptom. That is pretty much as scary as the maths will go in this talk, and we can use this quite a lot for applied purposes, but also for much more fundamental purposes. And one thing which we can show mathematically, prove, is that if you're really interested in disease, the genome is much less informative than the molecular phenotype. Most molecular biologists would have argued that anyway, but it is important to keep in mind as we try to personalize medicine that just providing people with the states of their SNPs is very, very poorly informative about many of the interesting diseases that plague us right now. So a brief outline about what I will talk about. I will touch upon population level challenges, which I think are fascinating, but I will only briefly mention them. Coming back to sort of more my home territory, system level challenges, briefly touching upon technology challenges, and then coming to statistics. Statistics is just a way of finding out how nature works in an organized fashion. It is not scary, it's terribly taught in most places around the world, but it's actually quite good fun once you understand the basics. And I will give a cartoon guide to some statistical developments. I won't touch upon the strengths that NICTA brings to the table, how ICT and health informatics can improve situation here. But there are exciting developments in telemetry, confidential information transmission and storage, integration of data that are best dealt by people from a strong ICT background, which I won't deal a lot with. I will much more try to touch upon problems such as heterogeneity, the evolutionary history, and cell decision-making process at the system level, and we come to that bit by bit. Before we start with that, it's important to keep in mind that if we want to alleviate disease burden globally, there are 3 easy interventions that would probably deal with most of the problems on a global basis. If we provide clean water, access to birth control, and preventative measures such as informing people and encouraging not to smoke, then these would probably have a much bigger impact than most of the other things I will talk about for the next half hour or so. So that is worthwhile to keep in mind. But, and that's quite a big but which only occurred to us recently, we thought that most complex diseases are limited or only of interest to what we would call fat white blokes. I think that is no longer the case because certain disease phenotypes, especially hypertension and drug response phenotypes, are well established to have considerable variation between different self-identified ethnic groups. For example, hypertension is vastly increased in some sub-Saharan African populations compared to European populations, and drug response phenotypes, for example, the response to warfarin, to codeine, or even to paracetamol, differ quite substantially between ethnic groups. And as societies become more industrialized and adopt Western lifestyles and diets, diseases that have been associated primarily with fat white blokes increase at alarming rates in other societies. This is now taken from a now 9-year-old review that Jared Diamond wrote in Nature, where he plots the incidence averaged across the populations and ages of type 2 diabetes in different population groups. And what emerges from this table is that the only people who can really afford to live on a Western lifestyle and diet are people of European descent. Because they, even in highly industrialized societies, have reasonably low incidence rates. That's averaged across ages, and that's a few years old now. But still, Australia or Germany a few years ago, 2% does compare very, very favorably with, for example, Asian Indians living in urban Fiji of 22%, or the Pacific Islands on Nauru, 41%. US Pima Indians are pretty much all full-time employees of the NIH now, which runs its own station outside the District of Columbia at the Pima Indian Reservation, Arizona, because of these high levels of type 2 diabetes. And that is becoming a huge problem in, for example, South Africa, India as well as various Pacific Islands nations. So that is something which we have to keep in mind, that complex diseases which we might have thought were limited to highly technological societies are creeping up all over the world, and there is a good ethical case as well to look at these things again. So at the population level, the clearest signals that we have so far come from pharmacogenomics. because pharmacogenomics is essentially basic biochemistry. We understand a lot about how these enzymes work. And we have ample evidence, for example, cytochrome P450 deals with pretty much any drug that you want to buy when you're ill is in some way or another metabolized through P450. Harbors lots of polymorphisms that influence how you metabolize drugs. ACE inhibitors are known to work much less well in sub-Saharan African populations than in European populations, and they work much, much better in Asian-derived populations. And BDIL, for example, was the first drug that was licensed to a specific ethnic group, so African-American individuals will receive this medication because ACE inhibitors do not work that well in congenital heart failure and hypertension. For warfarin, the clinical dosage that you might want to give a patient, these self-identified ethnicities are largely based on an atrocious scheme of ethnicities that the FDA used for a long time. So Asian is anything between the Arab Peninsula and Australian Aboriginals. That is all Asia, so including India, China, Japan, so it makes no sense whatsoever. But if you take this dose into account, then you will find that on average, the effective clinical dose of warfarin in an Asian individual is half the effective dose in an African-American individual. Hispanics, which is of an admixed population of African, Native American, and European ancestry, spans between the Asian and the white populations, and Caucasian individuals are sort of between the Asian and African individuals. And similar effects have been discovered and described extensively in the literature. Now, there are a couple of problems that arise from that. And first of all, is a good thing, is that we should be able to give safer drugs or give unsafe drugs in a safer way to different populations. So if you know this, we would just adjust our dose. And in the UK, that's routinely done by doctors now. But identifying such smaller groups where drugs would work also has huge implications for how industry sees drug treatment, because they suddenly see a fractured market where drugs work in smaller and smaller populations, and it's much harder to recoup any investments that you have made in the drug discovery. So that is a clear case where academia has to play a role first in identifying the populations in which drugs can be used safely and not safely, but also in trying to come up with better ways of addressing the need for drugs in smaller and smaller markets, and that will exercise us again. So that's the population level. This is just an illustration by David Goodsell, who is a professor for molecular biology at the Scripps Institute and also dabbles, or more than dabbles, in molecular art. So this is hand-painted watercolor of a receptor inside, of a nuclear receptor inside a human cell, and he paints everything at the resolution where you can see every single molecule at the level, at the size at which it would occur. You see DNA, RNA, and the proteins and the membrane of the nucleus. And we would like to describe that. There's no way that we would be able to describe such a mess, however beautiful it might look in the picture. There's too much detail, so we have to abstract from that in order to identify mechanisms by which we can interfere with this. So where might we want to interfere? And applications are obvious mostly in the sort of context of regenerative medicine. We would, for example, dearly like to be able to control the cell fate, how a cell decides whether it differentiates, proliferates, or undergoes apoptosis. We would like to understand the mechanisms by which it comes to these decisions, how non-random these decisions are, and how we can interfere with them. A simple example is the AKT signaling pathway. Here's just a hopelessly oversimplified representation. If you give epidermal growth factor, it goes to the epidermal growth factor receptor. Along a cascade, it will stimulate AKT. AKT will activate S6, and S6 will then lead to differentiation or proliferation under a host of different biological processes involved in insulin signaling, for example, but many other processes as well. So the question is now, how reliable is this information processing? This is just really only showing where information might flow. So EGFR, the receptor, does not only go towards AKT, but to a host of other proteins. AKT also stimulates more than just S6, and S6 has a myriad of different effects inside the nucleus. But also AKT is influenced not just by EGFR and its downstream mechanisms, but also by other influences. And finally, if we have a low and a high stimulus, can we map that to distinct outputs here at S6? And if we have an intermediate output, would the result of an intermediate input be intermediate to the outputs? So there's a host of phenomena which we don't understand, we would like to understand, and we are now starting to have a hope of understanding if we combine technology and modeling a little bit more. Here, the overarching problem in the literature is that if we just stimulate a biological system at single cell level, then the system does not behave in a nice, simple, straightforward fashion. It behaves, yeah, for lack of a better word, like a drunk person trying to walk in a straight line. It always goes to left, go to right, go to left, right, and so on. So there will be lots of fluctuation in, for example, the activity profile of S6 following identical stimuli, and different cells will behave differently. So rather than a fixed response, we have a distribution of responses. And the distribution of responses can be wider and thinner. We would quite like to control this, but first we have to identify where this variation comes from. This we can now do using mathematical modeling, and we can also validate this in experimental settings. And for example, for this type of pathway, serial transduction pathway, where this would be EGFR, this would be S6, we know that the degradation of S6 would contribute half the overall noise. So if we control this aspect of the noise, we would change the variability here. Equal results can be made for slightly more complex catalytic reactions and so on. And there are now ideas from fairly sort of advanced mathematical fields, theory of stochastic processes, that are beginning to have a direct and immediate impact on systems biology, on our understanding of how cells make these decisions. And increasingly, we have seen the first glimpses that we can actually muck around with cells and get them to do what we want in a dish, in cell culture. And there's something we're quite excited about. So let's have a look. So that's just at the cellular level. These in different cells, genetically identical cells, there will be differences. What happens now if we have a population of patients? And for example, we might have anything which is so vaguely red might be a disease-carrying individual, and you have empty, so you have gray circles and red circles. The red ones would be slightly more severe disease cases than these gray ones, but they would have similar underlying causes. And equally, you would have healthy people in blue, but the gray ones would be genetically with the gray filling would be genetically similar to those ones in red at some level or another. So in systems medicine, we have all the complications of systems biology, that identical cells can reach very different types of decisions. And we have to take into account that different hosts, different patients are also very different. So we're dealing with a bit of a problem. And how that manifests itself is still under intense investigation by a host of different groups around the world. We've been working for the past 2 years with Irene Roberts and Tassos Kara-Dimitris at the Hammersmith Hospital, who are neonatal and fetal hematologists. But they also look, especially Tassos, looks at grown-up populations of multiple myeloma sufferers. And here's some data from one of the largest cohorts of multiple myeloma patients that we have. And what we have done is we've quantified B cells, pre-plasma cells, a type of intermediate cell which I won't go into it, and plasma cells inside the bone marrow of patients and inside the peripheral blood. And we believe that the relative abundance of cells, this is a log scale by the way, is indicative of disease progression. We can treat them for some time, but they will all relapse. So far we haven't been able to cure anyone. So humanity hasn't been able to cure anyone. And there is a belief that knowing how many B cells, pre-plasma cells, and plasma cells you have will give you some information of how quickly the relapse to disease will occur. But what we find is each dot corresponds to one patient. In the patients, we have huge variability between patients, and equally in the bone marrow and in the plasma cells. If, however, we just divide the whole data up by patients, we get a slightly different picture, which still looks like a bit of a mess. But if we look at much, much more detail, what we have done here is we look at disease and recovery and at relapse and recovery in individual patients. And we find here these cell colors correspond to the different cell types. We find that in each patient, there is a quantitative but not a qualitative difference. So from disease to relapse, the abundances of these populations go down. So disease is clearly associated with a high level of all of those cell types, but you have many more PC cells. So we can, if we distinguish by patients, we can start to make some sense in that data, and we can actually start to apply some mathematical modeling approaches. And I won't go into detail because that's hopefully coming out reasonably soon in a nice journal. We can show that there is an unexpected transition from plasma cells to the pre-plasma cell stage. Now that's another case where, for convenience, medics and biologists have classified blood cells in a sort of hierarchy of distinct groups, where there seems to be, in fact, a much more of a continuum of different cell types. But it turns out that the best explanation for the change in cell abundances between recovery and relapse seems to be that plasma cells can go into this stage again, which then seems to be slightly more susceptible to various other factors that influence disease. In particular, you tend to have many more drug escape cells, mutants, in this cell pool than in any other cell pool. So this is one thing where even such raw patient data, so fairly low coarse-grained data, does allow us to gain some meaningful insights. So how much of these systems do we know? And we know very little. I use this in London a lot. I have a similar map of Melbourne, but I didn't want to show that. So it's just, if you want to look at the London Underground network and we imagine that everything is covered by clouds, and I tell you now you have to go from Euston to, let's say, to Heathrow here. How would you get there? If you know the underground system in London, you know you will be able to get there and you can reconstruct the path doing that. That's fine. But now let's go to a much more interesting city, my hometown Munich. Oh, not quite hometown, but anyway. So, and here you would be hard pressed to identify a way from one end to another end. And so what we have to find a way of doing, so getting rid of these clouds here and reconstruct the whole network. But when we do this, and that's why the Munich network is quite a good one, is networks are not everything. Networks are static description of something which is in fact highly dynamic. And in Munich, the nice thing is that, for example, here you have the zoo. The zoo is not apparent from the underground network. There is no zoo station in Munich, but there is a zoo marked, and here's a beer garden. So those are also things which don't have marks. And even, it's in fact a much better symbol because we have zoo and beer garden, but Munich isn't mapped correctly by just knowing this one zoo and this one beer garden, because there's in fact a much nicer beer garden just 400 meters away from this one here, which is even mentioned there. So all these network pictures should be treated with some level of skepticism. And we have made a rule that any statistic that does not work if you apply to the London Underground system surely cannot apply to the human protein-protein interaction network. So if you have a sanity check saying, if this is the protein-protein interaction network in humans, then you can't say anything about the protein-protein interaction network in humans. You can only say something about this small subset here. And so this is something which, if we could lift the clouds, and it turns out statistics give us a way of doing that, certainly for protein-protein interactions, then we can do this. And what we've done a few years ago is we looked at the number of genes in different species for which we had lots of protein-protein interaction data. And if you're if you're a physicist new to biology, you would feel slightly dented in your self-confidence if you see that you're just a couple of thousand genes richer than a worm. And so, but if you just apply pretty much the best mathematical techniques that you can apply and try to estimate the number of protein-protein interaction in these species, then you find that we beat worms something like, I have no idea, who cares really, but it's something 2 to 1, something like this. And still that is only just for the coarsest grained molecular processes. But we believe now, and that's a couple of lazy and a couple of good reasons, and the lazy reasons are in fact not so lazy reasons, but that it's a much better idea to look at protein-protein interactions and proteomic processes in general for diagnostics than at the genome sequence. Because, for example, here we have a protein-protein interaction between, I've forgotten the names of two proteins, but they were isolated from camels of all things. I just asked one of my students to find the nicest protein pair that he could find in the database, and he came up with something from camels. But if many biological processes are mediated by protein-protein interactions, and the protein-protein interactions are there in a very transient state, so they're there for a moment and then they're gone again. And we believe that interfering, messing up the ability of bad protein-protein interactions to occur is quite a neat way, and we have certain candidates that we believe might be reasonably nice. And that sort of mimics current trends in certainly the UK and the US to look at allosteric drugs as a replacement for targeting single genes. So we are quite optimistic at looking at these targets where rather than just one gene, you try to look at a set of genes, set of proteins as a target for drug treatment. How do we do this? And this is where I go into the slight science fiction area because I find this endlessly fascinating, the technology development. This is from a recent paper in Lab Chip from one of my collaborator groups from David Klug, which does single-cell proteomics and transcriptomics. With this technology, we can at the moment quantify up to 100 molecular species at single-cell resolution. We can do mRNA, protein, and phosphoprotein probes. So all these patents have been given as well. And what we can do is we just have single cells which we put into one of those chambers here. So in green, we have a single cell. And a microfluidic device where we can use optical tweezers to move the cells into these little compartments here. In the little compartment, we focus a microscope onto the cell, and we focus a laser some 10 micrometers above the cell. We then have a very intense laser pulse, which we shoot 10 micrometers above the cell. That will give rise to a cavity because of the heat that it generates. The cavity will expand, and the shear forces through the cavity expansion will burst the cell open. So it will just go through the cell membrane, and then we suck all this sort of lysate over a chip which is tagged with antibodies, and we can get very, very good quantitative assessment of mRNA and protein and phosphoprotein at the same time from the same cell. We can only multiplex this up to maybe 100, 200 or so cells, but we get much, much better quantification than at single cell level than we had thought possible. And so David Klug is quite uppity about his technology, so we get very nice results through that. Another nice result, where it's really just only a showcase of technology rather than anything else. Another nice result that we have is being able to image the bone marrow inside living organisms. So we know that hematopoietic stem cells in humans produce something like 1 million or 2 million new blood cells per second, every second of your life. You produce 2 million blood cells. But we don't know where precisely inside the bone marrow these hematopoietic stem cells reside. Do they live close to osteoclasts, osteoblasts, vasculature, and so on? So we need to map in vivo the bone marrow in order to identify where precisely these cells survive and live, because we believe that that has some impact on how we would treat other types of hematological disorders. And here's data from Christina Lucelso, a young lecturer in our department, who has developed a technique when she was with John Scadden at Harvard, where she can actually just image through the skull of a mouse. In blue, you see the bone. In green, you see osteoblasts. In red, you see the vasculature in 3D. So that's a living mouse. It's not a very happy mouse. It's anesthetized, but it's alive, and we can really see where inside these mouse skulls, in the mouse bone marrow, the stem cells live. So these technology developments are enormously exciting, but they still leave us with a problem that the data that we have is very detailed and very hard to interpret. So the final part is therefore, how can we make sense of that? And here we'll go slightly into sort of hopefully a user-friendly introduction to statistics. So we are now interested in using these technologies, or existing so much more standard technologies, to identify good ways of classifying diseases. One of the things that we are looking at is, together with Charles Bangham at St. Mary's Hospital, at human T-cell lymphoma virus, HTLV1. It affects something like 20 million people worldwide. It's a very, very close relative of HIV. In fact, HIV was first called HTLV3, not HIV. And in 2% of people, it caused a debilitating and horrible disease, ham-TSB. So I'm still at heart a physicist, so long names are abhorrent to me. So it's a horrible disease, and I can't remember what this acronym stands for. But it's enormously debilitating, and most of these people end up in a wheelchair and with severely compromised quality of life. So, but what we're interested in, what's the difference at the molecular level between people who develop this pathology and asymptomatic carriers of this virus? We have a fairly standard, some might say poor mass spectrometry data, because we use cell-deep mass spectrometry to analyze blood protein content from initially some 70 or so patients and a few controls with mass spectrometry, with HAM, with asymptomatic carriers, some multiple sclerosis, and then another sort of control group. And we have here now this set of peaks in our mass spectrometry spectrum, and we would like to identify peaks in such spectra that allow us to discriminate between people that are asymptomatic carriers and people that carry this terrible disease. First of all, we replace these spectra by discrete peaks. That's straightforward enough, but then we still have to identify which of those peaks correlates with one disease but not the other. What we're doing, we use something which is logistic regression. Those are details which shouldn't concern you at all, because these formulas here are quite harmless, really, but it's also reasonably dull. Each of those x's is a peak, and we are looking after the peaks. We want to see how important is the presence or absence of a peak in determining whether the person will develop the disease or won't develop the disease. So the x's are the interesting thing. And what we want to do is we want to identify as few of these x's as possible, but as many as necessary. So if we need 4 peaks, 4 proteins to identify if it's the disease or not, we take 4. But if we can get away with 3, we take 3. There are good reasons for that. We in total have, I think, 9 different cases we can look at. And we rank them by some sort of, you can treat it, if you want to, as magic. It's not quite, but it's as close as possible. We just rank the importance. So the earlier a peak appears here away from 0, the more important it is. So x2 is non-zero quite early on. So we have this parameter lambda, which we vary, and I won't go into this because the principle will become apparent without caring about the details. So the first thing that we would select as an explanatory variable between disease and non-disease cases is this protein x2. If we want to have 2 included, we get x2 and x8, and we go further and further and further until we have all our peaks included. So in total we have 10 peaks, so I got that wrong. So we clearly don't want to have all 10. We want to have the best set that we can choose. How do we assess statistical performance? We have one data set, and we now pretend that we divide the data set into 3 parts, 3 equally sized parts. What we do is we take this part here and now try to identify the best x's, the best proteins that would explain the data, that would allow us to distinguish between disease and non-disease. And if we find a couple of markers, we find that for 4 markers, we would get 19% misclassification. If we use this data set to train and these 2 blue boxes to test, we would get a false assessed rate of 20%. If we take the third set, we would get another regularization path, it's called, which tells us which ones to choose. We would get something like 20% again. So in total, we have tested our prediction approach, and we found by and large acceptable misclassification rates of about 20%. So the predictive performance of our approach is good. We are happy with that. But, and I think that summarizes one of the essential problems of personalized medicine beautifully. For the first data set, we used proteins X2, X3, X5, and X7. When we used the other data set to train, we identified X1, X2, X5, and X8 as the next sample. And when we did the third test, we identified X1, X7, X8, and X10 as the best predictors. So that means just going from one of those boxes to another box completely destroys the predictive performance of your predictors. So that means if we now take our 70 patients and we were to try to apply the same sample to the population at large, there's a fair chance that we would get it wrong as well. And one of the things that Meinshaus and Bühlmann have developed in 2010, remarkably late in my point of view, is that predictive power isn't everything. We want to have predictive power, but we also want to have stable, robust power. If we go to a new population sample, we still want to be able to identify disease correctly. And that's a fairly recent concept going by the name of stability selection. And here you strike a compromise between the predictive power and choosing the same type of biomarkers as often as possible. So you want to have the most robust set of biomarkers, which in a clinical setting is the one which you want to put in a test. Because those 4 or 5 biomarkers, if you put them in a test, you can give to doctors, and the doctors will be able to identify disease, hopefully with very, very low misclassification error, just because you had the wrong set of markers. And this problem of marker replicability is a big problem. I won't go into any of those details of how we do it, but we do much, much nicer than previously thought, and we are able to identify a robust and transferable set of markers. And we just use a very simple training thing. We just try to identify everything above this line will develop the disease, everything below this line will not develop the disease, and the crosses are mostly below the line, the dots are mostly above the line. So that gets us about, again, 20% or so misclassification rate. But at least we are fairly certain that in a new sample, these markers that we have chosen here would be very good. That's just based on 2 markers, by the way, not just on the 4 sets that we have chosen in the end. We can also identify that people who have MS lie solidly above also have differences in these 2 markers, and we can identify that MS is clearly different from asymptomatic carriers, but not that different from ham-TSP patients. And we went to a second set of asymptomatic carriers from Japan, I believe, where we found that they also, in a completely different population, we found that they also adhere to what we believe to be the case for asymptomatic carriers. So we're able to produce fairly robust and results that can be replicated between different populations. To summarize and maybe to proselytize, here, so some sort of conclusion. So what we want to do is we want to collect all the data that is informative and treat it with the value that it deserves. It's a common mistake to assume that all data are good data. That is absolutely not the case. Some data will contain no information whatsoever. Some data contain information, but you have to be careful how you interpret it. Data fusion tries to draw inferences from such diverse, often noisy, partially contradictory data in a unified framework. And we believe that these sort of tools, stability selection, something which I haven't touched upon, elastic nets, and non-parametric Bayesian methods, allow us to do a very nice job at taking only the useful information from the data without amplifying any noise. Now, this is purely pattern recognition, what we've done here. We haven't looked at mechanisms. All we can do from this type of approach is we can then identify candidate mechanisms, very much like we can use these proteins that we have identified as being predictive for ham-TSP versus asymptomatic carriers, and we have to follow that up. But we can do a very nice job, and if you think about, most doctors of a certain age, or certainly, let's say in the UK, most of the consultants that you would go to with your cert, would admit that a large part of their job relies on pattern recognition. They have seen all sorts of malignancies before, they're able to classify them quite precisely. At this level, we only want to replicate years of experience in a doctor using statistical learning approaches. Ideally, we would like to go beyond that and also gain some understanding into these mechanisms. So these approaches, therefore, have to be backed up by mechanistic modeling, where we really try to identify or formulate mechanistic model hypotheses about how these things actually work. And you have to combine those two frameworks together. That's the cartoon guide to statistics. XKCD is a comic, an online comic, and it works beautifully as a sort of introduction to statistics. So if you can't read it, this lady, it's all about stick figures, so it is not the most sort of detailed depiction of reality, but anyway, statistics are fairly accurately described. So this lady comes to the man. This is all crudely done by hair, obviously, in this case. So jelly beans cause acne, scientists investigate. So she says that, and then they look at the study, and they tested between jelly beans and acne was no correlation. Then they look at the different colors. We look at blue, brown, and so on. And finally here they find there's a link between green jelly babies and acne. The hypothesis test now has a p-value of greater than 0.05. So that means you can reject the null hypothesis that there's no link. So you would think. But what you have done is you've generated the same test several times, whatever, some 20 times here in this case. And so you can't use the p-value for a single test. The more often you test for something unusual, the more likely it is that something unusual will happen to you. And that is the basic story that we have to keep this in mind. So I think that's something which is very important to remember as we get more detailed data. And there's a real danger in some parts of personalized medicine where some companies start to peddle chips that would allow you to test however many SNPs and give you an idea of what disease you might have, or other markers, or that some private practice in the United States, for example, give you preventative checks every year, which include CTs. The radiation that you incur as part of a routine CT test, if you do that every year, you're essentially nuts. I think you have a much, much higher chance of developing cancer just because of the CT scan than anything else. So there is some danger, and these sort of statistical issues, and particularly this multiple testing one, are quite important. Another important example is that recently one of the present government politicians in the UK said that no hospital should be worse than average. This is also a level of statistical lack of numeracy, which is slightly worrying, and is important to keep these things in mind. So to finally conclude, the more we target healthcare individuals, the less statistical support for therapy we have. So if you look at each individual person, there's no control group against which you can compare this person, unless it's an identical twin. But then we only have one and one comparison. So we have to have different statistical and computational tools. And there, incidentally, the data integration, being able to tap into the data that one practice, for example, in Brisbane has collected, and using that in Melbourne or in Perth is quite important, that you can have tools which allow you to draw on experience from other doctors and increase the ability to perform careful pattern recognition. So for the foreseeable future, I think we will require a mix of interventions based on mechanistic understanding or just simple experience based on pattern recognition. But both of those require a little bit of care and sort of considerate statistical and computational nous as you apply these approaches. And the important thing for us is that these new technologies that we are developing, which give more detailed data at single cell level and so on, in fact, don't get rid of the need for statistical methods, but increase the need for such methods. And is actually a very exciting field to be in. On this note, I will close with brief thanks to the people in my group who have carried out the work and the funding bodies who have funded it. Thank you very much.
Before we thank Michael, I'd like to thank our sponsors, Melbourne School of Engineering, the Centre for Neural Engineering at the University of Melbourne, Monash University, including Monash Biomedical Imaging, RMIT University, the Melbourne Convention and Visitors Bureau, Phillips, Ormond Fitzpatrick, IBM, the Victorian Life Sciences Computation Initiative, the Bonics Institute, the Florey Neuroscience Institutes, the Bionic Vision Australia, the Bio21 Cluster and NICTA. So would you please join with me in thanking our sponsors for making this event possible. Thank you. And of course, reserve our special thanks for Michael and his informative and illuminating presentation, and we'll look forward to seeing you back in Melbourne at some further stage, Michael, and have a safe trip home. Please join me in thanking Michael Stumpf. Thank you, Michael.